Even before microorganisms were seen, some investigators suspected their existence and responsibility for disease. The Roman philosopher Lucretius and the physician Girolamo Fracastoro suggested that diseases was caused by invisible living creatures. In 1665, the first drawing of a microorganism was published in Robert Hooke's Micrographia. However, the first person to publish extensive accurate observations of microorganisms was the amateur microscopist Antony van Leeuwenhoek. Antony van Leeuwenhoek's microscope could magnify around 50 to 300 times, and he may have illuminated his liquid specimens by placing them between two pieces of glass and shining light on them at a 45-degree angle to the specimen plane. This would have provided a form of dark field illumination in which the organisms appeared as bright objects against a dark background and made bacteria clearly visible. In 1673, Leeuwenhoek sent detailed letters describing his discoveries to the Royal Society of London. It is clear from his descriptions that he saw both bacteria and protozoa. Microscopic observations of microorganisms do not provide sufficient information to understand their biology. For the discipline to develop techniques for isolating and culturing microbes in the laboratory were needed. Many of these techniques began to be developed as theory of spontaneous generation. This studies on the role played by microorganisms in causing disease ultimately led to what is now called the golden age of microbiology. From earliest times, people had believed in spontaneous generation, that living organisms could develop from non living matter. Even Aristotle thought some of the simpler invertebrates could arise by spontaneous generation. This view finally was challenged by the Italian physician Francesco Redi, who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. Reddy placed meat in three containers. One was uncovered, a second was covered with paper, and the third was covered with a fine gauze that would exclude flies. Flies laid their eggs on the uncovered meat and maggots developed. The other two pieces of meat did not produce maggots spontaneously. Flies were attracted to the gauze covered container and laid their eggs on the gauze. These eggs produced maggots. Thus, the generation of maggots by decaying meat resulted from the presence of fly eggs, and meat did not spontaneously generate maggots as previously believed. Similar experiments by others helped discredit the theory of larger organisms. Leeuwenhoek's discovery of microorganisms renewed the controversy. Some proposed that microorganisms arose by spontaneous generation, even though larger organisms did not. They pointed out that boiled extracts of hay or meat would give rise to microorganisms after sitting for a while. In 1748, the English priest John Needham reported the results of his experiments on spontaneous generation. Needham boiled mutton broth and then tightly stopped the flask. Eventually, many of the flasks became cloudy and contained microorganisms. He thought organic matter contained a vital force that could confer the properties of life on non living matter. A few years later, the Italian priest and naturalist Lazzaro Spallanzani improved on Needham's experimental design by first sealing glass flasks that contained water and seeds. If the sealed flasks were placed in boiling water for 3 by 4 of an hour, no growth took place as long as the flask remained sealed. He proposed that air carried germs to the culture medium, but also commented that the external air might be required for growth of animals already in the medium. The supporters of spontaneous generation maintained that heating the air in sealed flasks destroyed its ability to support life. Theodor Schwann allowed air to enter a flask containing a sterile nutrient solution after the air had passed through a red hot tube. The flask remained sterile. Georg Friedrich Schroeder and Theodor von Dust allowed air to enter a flask of heat sterilized medium after it had passed through sterile cotton wool. No growth occurred in the medium even though the air had not been heated. Despite these experiments, the French naturalist Félix Pauchet claimed in 1859 to have carried out experiments conclusively proving that microbial growth could occur without air contamination. This claim provoked Louis Pasteur to settle the matter once and for all. Pasteur first filtered air through cotton and found that objects resembling plant spores had been trapped. If a piece of cotton was placed in sterile medium after air had been filtered through it, microbial growth occurred. Next, he placed nutrient solution in flask, heated their necks in a flame, and drew them out into a variety of curves while keeping the ends of the necks open to the atmosphere. Pasteur then boiled the solutions for a few minutes and allowed them to cool. No growth took place even though the contents of the flask were exposed to the air. Pasteur pointed out that no growth occurred because dust and germs had been trapped on the walls of the curved necks. If the necks were broken, growth commenced immediately. Pasteur had not only resolved the controversy by 1861 but also had shown how to keep solutions sterile. The English physicist John Tyndall dealt a final blow to spontaneous generation in 1877 by demonstrating that dust did indeed carry germs and that if dust was absent, broth remained sterile even if directly exposed to air. During the course of his studies, Tyndall provided evidence for the existence of exceptionally heat-resistant forms of bacteria. Working independently, the German botanist Ferdinand Kohn discovered the existence of heat-resistant bacterial endospores.